Usually when you sign like a middle school science project, there's this unspoken expectation of edible art.
Oh, definitely. The classic middle school trope.
Right. You give an assignment on the cell, and a few days later, a student walks in proudly holding a baking tray, and inside is the dreaded cell cake.
A massive just slab of frosting.
Exactly.
You've got like a jawbreaker for the nucleus, squiggly gummy worms for the endoplasmic reticulum, and it just sits there. It's colorful, sure, and sticky, but completely static.
And it forces a really uncomfortable question for us as teachers. Are we grading their scientific cognition, or are we, just grading their baking skills?
Most of the time, it's the baking skills.
Yeah.
Yeah.
So today, we're tearing down the cell cake. Welcome to our deep dive. And we are speaking directly to you, the middle school science educator.
Specifically, those of you building 7th grade life science instruction.
Yes, instruction that's aligned to the NGSS, the Next Generation Science Standards, and the Alaska Science Standards.
Right. And to do that, we are operating today from a sort of conceptual space that we call the Bioforge Educator Lab.
I love the aesthetic of this space.
It's great, right? Picture a solar punk field laboratory. It's bathed in this really rich, warm, golden light.
Yeah, and there are brass drafting tools just scattered across these wooden workbenches. You've got transparent overlays of ecosystems, wireframe models of DNA.
And these lush living terrariums sitting right next to really intricate cell diagrams.
And the dominant color of this entire conceptual space is a warm golden brass, like hex code B88A2E, if you're into that sort of thing.
Which represents the really rich foundational nature of the science practice we are unpacking today.
Exactly. Developing and using models. That is our mission today. We want to help you transform models in your classroom from like static art projects into dynamic, rigorous thinking tools.
Because we really have to figure out how to move past the icing and the salto dioramas to get to the real cognitive work.
Right. So to move away from that cell cake mindset, I feel like we first have to completely redefine what a model actually is.
Yeah, and more importantly, what it does in a modern science classroom. Because a scientific model is a representation used to understand or explain, predict, or communicate how a system works.
It's not just a picture.
No, not at all. The research literature actually refers to this as an epistemic object.
Epistemic, meaning?
Relating to knowledge or knowing.
Right.
It's a tool for figuring things out. It allows students to map out the parts, the interactions, inputs, outputs, boundaries, processes, all of it.
So it's really not a replica. The goal isn't just to make a miniature version of the real thing. It's more like a map of mechanisms.
Exactly.
Yeah.
Which honestly requires a massive paradigm shift for a lot of educators and students for that matter.
Also.
Because a model is always incomplete. Always.
Okay, wait. I have to push back on that a little bit.
Go for it.
Because I'm thinking about the realities of a 7th grade classroom, right? Let's say a student is building a model of a cell, and they completely leave out the mitochondria. Or, I don't know, they're mapping an ecosystem, and they forget the decomposers entirely.
Right.
If they're learning this for the first time, won't an incomplete model just teach them bad science? I mean, shouldn't we be striving for perfect accuracy?
Well, here's the thing. If a model was perfectly accurate to reality, it would be as complex and incomprehensible as reality itself.
Oh, wow.
Models are abstractions. They are specifically designed to highlight certain mechanisms while intentionally ignoring others. That's how we make sense of complex dynamics.
So leaving things out is a feature, not a bug.
Exactly. Think about a subway map.
Okay.
A subway map doesn't show you every tree on the street above, or the potholes, or the exact geographical curves of the underground tunnels. It leaves all of that out so you can understand the system of stops and connections.
Right, because if a subway map was perfectly accurate to the actual geography, you wouldn't be able to read it to get from point A to point B. It'd be a mess.
Precisely. And that makes a lot of sense, especially in life science.
Yeah, because so many of the systems we teach just cannot be observed directly.
They're either deciding what parts and relationships actually belong in this specific model.
Okay, so picking the stops on the subway map.
Exactly. Then step 2 is explain. Articulating what this model helps us understand.
Which is a huge shift, right? Moving from just naming things to actually explaining mechanisms. Instead of just pointing saying this is a plant, it becomes this represents how energy from the sun is captured.
You hit the nail on the head. Then comes step 3, which is where the real cognition happens. Test.
Like a quiz?
No. Test the model. Does the model match the evidence we have?
Oh, I see. I think of this step like Like drawing a blueprint for a house, building the plumbing, and suddenly realizing you put the pipes in backward.
Right.
The water's flowing into the living room instead of the sink. You run the simulation in your head or on paper, and the system breaks.
And when the system breaks, you move to step 4, revise.
So fixing the plumbing.
Exactly.
Hmm.
Based on the new data or the broken simulation, what needs to change in the model?
Okay, so if a student's initial ecosystem model shows energy flowing from the rabbit to the grass.
Testing that against observational data reveals the backward plumbing. Rabbits eat grass. Grass doesn't eat rabbits.
Right, so they literally just cross things out or use a different colored pen to draw a new arrow.
Yep. And finally, step 5 is communicate, where they use that newly revised model to help someone else understand the system.
Okay, so representing, explaining, testing, revising, and communicating. Applying this 5-step cycle to specific 7th grade examples really changes everything. I mean, it means we don't need the cake anymore.
We really don't. We can use everyday objects to map structure to function.
Give me an example from the sources.
Well, there's a great one for the cell as a system. Instead of frosting, use a simple plastic bag.
Okay, a plastic bag.
Yeah. Instead of just memorizing the word membrane and labeling a circle on a worksheet, students use the plastic bag to represent the boundary.
They might call it the border patrol of the cell.
Right. And then they use a large storage bin to represent the vacuole. They are physically manipulating these objects.
And that physical manipulation is key, isn't it? It creates a tactile memory of boundaries and containment.
Right.
The bag isn't just a flat circle on a page. It's a physical barrier that keeps things in and out.
Exactly. And that same tactile principle applies to food webs, too.
Oh, yeah. I've seen teachers use a string web activity for this.
The string web is fantastic.
It really is. You have students stand in a circle. Someone is the sun, someone is the grass, someone is the rabbit, the hawk. And you connect them all with a ball of string.
And then you introduce a disturbance to the system.
Like a drought.
Right, a drought kills the grass. So the student representing the grass drops their piece of the string.
And the psychology behind why that works so well for a 12-year-old is fascinating. Because the student playing the rabbit physically feels the slack in the line.
Yes.
And then the hawk feels the tension change. It makes systemic collapse completely visceral.
Compare that to a textbook diagram of a food web.
Oh, it's night and day. A textbook with some arrows pointing left and right doesn't communicate the immediate compounding danger of an ecosystem collapsing. But the physical tension of the string does.
And from that visceral feeling, they are so much better equipped to move to graphic models on paper. They can map those inputs and outputs with a real understanding of the consequences.
Does that physical representation extend to body systems too?
Oh, absolutely. When modeling interacting subsystems, a student might model how, say, touching a hot stove sends a signal through the sensory receptors in the nervous system to the muscular system.
Causing the hand to pull away.
Right. The model has to show the input, the pathway, and the output.
Okay, but physical objects like string and plastic bags, they kind of hit a wall when we start talking about phenomena that completely defy our normal perception of time and space.
Like what?
Like, how does a student map out an evolutionary process that takes 10,000 years?
Ah, yes. That brings up a massive challenge. Physical tension works great for immediate cause and effect. But for abstract concepts, we have to look beyond 3D objects and drawings.
So the toolkit has to expand.
It does. It has to include digital simulations, graphs, and abstract system maps.
The sources introduce an approach for this called object process methodology, which, I'm not going to lie, sounds highly technical.
It does sound like a college textbook, but it really just boils down to mapping the nouns of a system against the verbs.
I love that concept so much because a middle schooler can easily grasp nouns and verbs.
Right. You force them to identify the object, the noun like a sugar molecule, and the process it undergoes the verb like cellular respiration.
By linking the nouns and the verbs, you completely bypass that trap of students just labeling things and calling it a day. They have to show the causation.
This approach is vital when we look at things like genetic variation and heredity.
Okay, back in the Bioforge, I'm imagining a gene conservatory section.
Perfect. In that space, you could model how DNA codes for traits using physical wireframes, or maybe ribbons where different color sequences represent different traits.
So you can physically show how traits are inherited and mapped on a family tree.
Right. But when you move to natural selection over generations, Physical models really struggle to demonstrate population-level shifts.
because it's too slow.
Exactly. This is where digital tools become absolutely essential. You need to compress time.
A virtual lab is essentially a volume knob for variables, right?
That's a great way to put it.
It's not just a time machine letting them see the 1,000 generations of bacteria in 3 minutes.
Yeah.
It's a control deck. A student can literally crank up the temperature in a virtual environment and watch, in real time, how the frequency of a trait-like temperature resistance changes across a whole population.
Or they turn down the moisture variable and watch the population adapt or crash.
So they're actively testing and revising their mental model of evolution by manipulating that digital model.
Exactly. They observe the inputs and the boundaries. Another abstract concept that requires this level of active manipulation is matter cycling in ecosystems.
Because you can't see atoms rearranging themselves during photosynthesis.
No, you can't, but you can track those unseen fluxes of energy and matter.
Using colored beads, right?
Yeah.
That's a classic method from the sources that just completely changes the cognition.
It really does. You use, say, 6 black beads for carbon, 12 white for hydrogen, 18 red for oxygen.
And students physically snap them together to make carbon dioxide and water molecules.
And then, this is the crucial part, they have to break those exact same beads apart and rearrange them to build glucose and oxygen.
They're acting as the mechanism. They become the verb in the object process methodology we were talking about.
Yes. Because they're the ones breaking the beads apart and putting them back together, they physically feel the conservation of matter.
They didn't gain any beads from the desk, and they didn't throw any away. They just rearranged what was already in the system.
They experience the boundaries and limitations firsthand. They aren't just memorizing a chemical equation on a whiteboard. They are proving to themselves that energy and matter flow into, out of, and within the system.
Now, all of these examples,
Mm-hmm.
the volume knobs of virtual labs, the colored beads, the string tension, they sound incredible in theory.
God.
But we have to ground this in the chaotic reality of managing a classroom full of middle schoolers.
Fair point.
These activities can so easily devolve into unstructured playtime if we don't have guardrails. So let's open up the educator's toolkit from the sources and look at the hazards in the lab. What are the instructional practices we need to avoid to maintain the cognitive rigor?
Well, the primary hazard is treating models as posters or decorations.
Okay, explain that.
If the model is just wallpaper for the hallway, it is no longer an epistemic tool. It has just become a presentation of facts.
Which ties directly into that trap we talked about earlier, grading artistic skill instead of scientific thinking.
Yes. A messy, scribbled diagram on notebook paper that accurately shows cause and effect, where a student has crossed things out and redrawn arrows based on evidence.
That is vastly superior to a beautiful, color-coded diorama that completely misses the core mechanism.
Exactly. And assessing their fine motor skills instead of their science. That is an equity issue, too.
Absolutely. What's another hazard?
The most counterintuitive hazard for many teachers is the urge to give students a finished model too early.
Oh, I can hear the pushback on that already.
I know.
Doesn't giving a 7th grader a blank slate just cause intense anxiety? I mean, if they are trying to figure out photosynthesis, shouldn't I show them what a good model of photosynthesis looks like first, so they know what they're aiming for?
It's tempting. But if you hand them the completed diagram at the start of the unit, you've robbed them of the cognitive friction required for sense-making.
Oh, that's a good phrase. Cognitive friction.
Right. You've turned them back into passive receivers of information. The struggle of trying to figure out where the arrows go, that is the learning process. The model isn't the final product. It's the rough draft of their thinking.
But when they are in that rough draft phase, we also have to watch out for cognitive overload. The sources emphasize not overloading students with too many symbols, arrows, colors, and vocabulary terms all at once.
A 12-year-old's working memory can only hold so much.
If they're trying to color code 8 different organelles while also drawing 3 different types of arrows for energy, matter, and information, the model just becomes unreadable to them.
They lose the mechanism in all that noise. And when that happens, they will often revert to the easiest task, which is labeling.
Right. Pointing to a mitochondrion and writing mitochondria is safe. It doesn't require explaining a process.
But we cannot let labels replace explanations.
Okay, so those are the hazards. But knowing what not to do isn't really enough when you're walking around the room and a student is staring blankly at a piece of paper, or just drawing pretty pictures of leaves.
Yeah, that happens.
How do we intervene without just giving them the answer? This is where specific teacher language prompts become your absolute best tool.
When you walk the room, your questions should shift them from passive memorization back to active cognition.
Give me some examples.
So if a student draws a shape, you simply ask, what does this part represent?
Force them to define the noun out loud.
Exactly. Then point to the empty space between two parts and ask, what interaction are you trying to show here?
Force them to define the verb.
You've got it. Another great prompt is, where is matter, energy, or information moving?
Oh, that specific prompt is brilliant because it hits directly on those cross-cutting concepts, the big universal ideas like energy flow and matter cycling that apply to everything from a single cell to an entire biome. It forces them to trace the invisible lines in the system.
And when they draw one of those lines and make a claim in their model, you challenge the foundation of it. You ask, what evidence supports this part of the model?
Okay, so if they draw an arrow showing plants getting their food from the dirt,
You ask for the empirical evidence. And when they review their lab notes and can't find any evidence that the soil mass actually decreased...
they realize the plumbing is backward.
Exactly.
And then you can hit them with the ultimate metacognitive question. What does your model leave out?
Yes, asking a seventh grader to identify the limitations of their own creation forces them to realize that their drawing is just an abstraction. It's a tool they built for a specific job, not reality itself.
To connect their specific tool to the broader system, you could ask a predictive question, right? Like, what would happen if this part of the system changed?
Perfect. If you remove the primary consumer from this web, what happens to the energy flow?
And when they realize the whole system shifts, you ask the final question of the iterative cycle. What should we revise based on this new evidence?
Those prompts completely change the power dynamic in the classroom.
They really do. You aren't just standing at the front dispensing facts to be memorized. You are walking around the Bioforge Educator Lab, guiding their sense-making. You are helping them refine their epistemic tools.
By shifting the goalpost from, I memorized the parts for Friday's test, to, I can explain how the parts work together using evidence, you are equipping your students with a framework they can use to decode the entire world around them.
That is the authentic scientific practice the NGSS and the Alaska Science Standards are demanding.
Exactly.
In this environment, the model is never the end product. It is the vehicle. We've established today that the best scientific models are intentionally incomplete. They leave things out like the exact shape of a subway tunnel or the dirt in a photosynthesis diagram so that the user can focus clearly on the mechanisms that truly matter.
And I think this raises a really provocative question for your own professional practice.
Let's hear it.
If a scientific model is intentionally incomplete so that we can focus on what truly matters, what outdated instructional practices might you need to leave out of your own pedagogical model?
Oh, that's powerful. Do you need to leave behind the cell cake? The vocabulary matching worksheets? What do you need to erase from your own instructional blueprint so that your students' real cognition can finally shine through?
It's a lot to think about.
It is. Take some time to think about what you are going to leave out of your next unit. Thanks for joining us for this deep dive into the practice of modeling. We'll see you next time in the Bioforge.